Hello friends, welcome back to my YouTube channel Nastic Badasari. So this video is regarding some questions that has been released in the particular response sheet as well as answer key. It is really confusing. So in this video I am going to share with you few questions which are confusing, and from CSIR, from NTA, definitely those questions will be considered as a drop, and many students who wrote those questions will be getting a grace mark. Okay, so please watch the video fully, only then you get to know an idea what are the questions that you can challenge and what are the questions that you should not challenge, because many students will be challenging that question without having much ideas. In this particular video I am going to discuss you what are the questions that you can challenge and for what questions you can able to get a particular grace mark. So first question was question number 9. So histone variants play important role in chromatin functions in mammalian cell. Which of the following correct in context of histone variants? So from official answer key they just mentioned histone variants have been reported for H3, H4, H2A and H2B. Okay, so this is already given in the particular thing. So you can refer this particular article. In that they had given a detailed explanation with regarding to histone variant. They just listed out the histone variants for H3. There is a variant, so H3 variants they just mentioned, H4 variants, H2B variants and H2A variants. So definitely you can't able to challenge these questions. Okay friends, the correct option is histone variants have been reported for H3, H4, H2A and H2B. So next question is question number 17, that is in page number 66. So this is question number 17. So this is the question number 70 that is asked from developmental biology topic. So in which one of the following developmental fate, the fate of maternal somatic cells is determined first, which further determines the fate of developing embryo? The correct answer is the specification of the dorsoventral polarity or dorsoventral axis in the Drosophila. So for which you can either refer the Gilbert book, which has given us sufficient information with regarding to that particular thing, and in addition to that you can also refer this particular paper friends. In it just mentioned the maternal control of Drosophila dorsoventral body axis. So please don't challenge these questions. The correct answer is the specification of dorsoventral axis in the Drosophila. So the next question that this is confusing by many students, is question number 27. That is, observed monosaccharide in the intestinal epithelial cell exit via which of the following transport? They just say, they didn't ask which particular transporter is involved in observing monosaccharide. They are asking by utilizing which particular transporter. So only if you read the question completely you can able to give you a correct answer. So for which you can refer this particular book chapter friends. In this book chapter they had given a clear explanation. And monosaccharide, that is 3 monosaccharide ones, all 3 monosaccharide, once they enter the enterocyte, then they will be exported out from the cell into the portal circulation. And the transporter which play an important role during this process is this GLUT, that is glucose transporter 2. Okay, so this is a low affinity facilitative sugar transporter, and many students are thinking this particular GLUT5 is the correct answer, but please don't think. GLUT5 play an important role in the fructose transportation. Okay, so fructose they are not transported using this SGLT1. Its entry from lumen into the observing cell is via this GLUT transporter. But here they are asking which particular transporter play an important role in bringing this observed monosaccharide in the intestinal epithelial cell. Okay, so if you read the question completely then you can able to give a correct answer. So please don't challenge this question. Even though if you challenge they are not going to accept your claim. Okay, so this particular answer GLUT2 is the correct option. So the next question is question number 43. So please regret my voice is not thing, because I have few health issues, that's why my voice is not clear. Sorry for that friends. So many students have demanded the challenging for this particular question. So please don't challenge this question. This question is given a correct option. A movie that featured dinosaurs should also have which of the following combinations of geological organism? Choose the correct option. So early diverging angiosperms, reptiles and amphibians, they are the correct option, because when a dinosaur has been lived in a Jurassic period, that period only this angiosperm, reptiles, amphibians were there. There was no human, there was no apes, and also before only angiosperms has developed first, and then only gymnosperms has been arise. Okay, so you can't able to challenge any other options apart from this particular option. Next particular question is question number 56. So this particular hemagglutinin protein in influenza virus contains a long alpha helix with a 53 residues. Which are the following describes attributes of alpha helix? I don't collect any references with regarding to some other things, but then in this particular paper they just mention the particular size, that is they just mention the length of the alpha helix is 75.6 Armstrong, but it is 80 Armstrong. Okay, so almost it is similar. So this particular thing only I just identified, and I don't think so there are evidences we cannot able to identify this particular option is incorrect. This particular option might be correct because I don't able to search many research article, or even in Lellinger book they didn't speak about this particular thing. When I just searched the recent article with regarding to the structure of alpha helix in hemagglutinin protein, so I couldn't able to get any answer. If any of you are stating this particular option is incorrect, or if you are going to challenge this question, please do tell me in the comment section so that it will be very much helpful for those students. Okay. Next question is question number 65. The initiation of transcription. That is, there was a question with regarding to initiation of transcription. So they are asking with regarding to the abortive initiation event. Okay, so for which they had given this much options have been given. So many students are asking many to challenge this particular question. The correct option for this particular question is they had given it is B, that is it is A and C is the correct option, like that they just mentioned, friend. So question number 65, it is 257 is the correct option. That is, promoter escape in bacteria is usually accompanied by the release of sigma factor from RNA holoenzyme complex. That's the correct option. Next, promoter escape in eukaryotics accomplished by phosphorylation of RNA polymerase large the C-terminal domain, that is carboxy-terminal domain. So while discussing the answer key, that is unofficial answer key itself, I just made you an enough amount of explanation with regarding this carboxy-terminal domain, how it has been phosphorylated and what are the effects of phosphorylation. In addition to that, we also discuss about this promoter escape pathway, everything I just read. So either pass this video and you can be able to know about all those things, friends. Okay. So coming back to the point, so this particular option is also correct, whereas option number B, that is abortive initiation even in prokaryote results in a formation of short transcript, that is 10 nucleotide. Yes, it is a correct, but in eukaryotes they just mentioned transcripts are approximately 75 nucleotides in length. So I don't think so this particular option is correct. That's why they had given. So here the question is that which of the following combination is correct. Okay, so ultimately option number A and option number C is correct. Why they are saying D is incorrect means they just mentioned promoter recognition in bacteria is governed by sigma factor, which will be binding to 10 and 35 regions of the particular DNA complex. Okay, this is correct, but they just mentioned this is regulated by RNA polymerase 3. Here they are speaking about the transcription initiation is bacteria. Bacteria have only one RNA polymerase, whereas RNA polymerase 1, 2, 3 are involved in eukaryotic transmission. Okay, so that's why this particular option is incorrect. Okay, so please don't challenge this particular question. The option is correct. At the same time, the question, there was no mistakes in any question. Okay, next question was 63 question. Okay, so this particular definitely those students who had attempted this question definitely will be getting a mark. So I think this particular question is belong to part C, I think so. So you'll be getting 4 mark, I think so. Okay, I don't think it's a part C question, friends. So definitely will be getting 4 mark. Okay, so when you are calculating raw mark, just add up 4 mark. Okay, so here this particular question they just mentioned 252 is the correct option, that is B, C, D. This is with regarding to excision repair system. So here they just in the Nature article also you can refer, and at the same time you can also refer Watson book, friends. So here they just mentioned the strand is it consists of 15 to 13 nucleotide fragment of damaged base will be there, and this will be released from the DNA duplex by a DNA helix called UVRD. But here what they just mentioned, the nucleotide range, that is here they just mentioned the size of the residue is 50 to 60, but it's not a long stretch of DNA. It is only a short stretch of DNA consisting of, so it's a short stretch of DNA which will be consisting of so 12 to 13 nucleotides. Okay, friends, so you will be getting a mark for this particular thing. Rest of the options are correct. The gap is generally filled by typically DNA polymerase 1, and the distortion caused by lesion is recognized and bound to UVRA and UVRB complex. So definitely for you, you will be getting this particular option. I think many will be getting, because this is the easiest repair system and many will be attempting these questions. Okay, so don't worry, for you definitely this particular question will be given a correct option. Next, many students are demanding for this particular question. Of course, they are asking incorrect statement like that they were asking, and this is a 4 mark questions. Yes, it's a 4 mark question. So the correct option, that is incorrect option, is that they contain a large quantity of sialic acid which inhibit the adhesion of charge weight repulsion contribution. Of course, IgG, so Ig superfamily consists of the sialic acid in the cell surface receptor. They are also playing this particular sialic acid is based on charged base repulsion. But here they just mention this particular sialic acid is inhibiting the cell adhesion, but that's not a thing. Nowhere in any research article they didn't mention about this role of sialic acid. It mentioned the sialic acid is inhibiting the cell adhesion like that, and all I didn't study. Okay, so this is not ultimately. So you can't able to challenge this question. The correct option, it is an incorrect one. Okay, so large quantity of sialic acid which inhibit adhesion molecule like that they been given. So they are asking from question, they are asking incorrect. So please read the question correctly. That's what I just mentioned you. Next is question number 75. Okay, so this was a 75 question. This is with regarding to this forelimb formation. So for which you can refer this Gilbert book, friends. So in Gilbert book they just mention you clearly about this particular thing. So in Gilbert page number, so it is page number 516, that is 516. You can able to have an entire description of what are the question that has been asked, friends. So in this book, in this particular paragraph alone, they were asked this particular question. So if you check with the question, loss of TBX5 gene results in complete failure of forelimb formation, and here they are asking which of the following statement is correct. Okay, so option number A and option number B are correct option. That is, loss of TBX5 gene results in complete failure of forelimb formation, as well as the hind limb bud growth and the initial patterning will be normal even though this TBX4 gene is knocked out, because this TBX4 gene's function will be replaced by TBXX5 gene. Okay, and here the rest of two option, misexpression of PIT-X1 gene in forelimb ceases the development. It says that if you misexpress this particular PIT-X gene, they says that the development of muscle, bone, tendon will be stopped. But this is absolutely a wrong, incorrect thing, friends, because if you check over here, the same statement is being given, friends. Misexpression of this PIT-X gene in mouse forelimb result in the transformation as well as translocation of muscle, bone, tendons. Okay, so but in the option they just mentioned stop. Okay, so that's why this particular option has been given a incorrect one from NT itself. Next, when islet 1 is inactivated, specifically in the lateral pit mesoderm, the hind limb still develop. Okay, so if you want to know in-depth analysis about this particular option, you can refer this particular paper. So in this paper, they had clearly mentioned about this particular islet gene 1. Okay, so if you inactivate this particular islet 1 gene, it will be causing a loss of posterior structure. So in the distal as well as proximal region, that is, it will be losing a hind limb. But what is the option that they had given over here? They said that when you inactivate this particular gene, the hind limb still develop. That's why these two options are given incorrect. Okay, so this option is also not being challengeable. Okay, so you can't able to challenge this particular option. Next is this particular 76th particular question. Okay, so question number 76. Yeah. So to those students who are attempted this particular question, definitely the mark will be there, friends. So please don't lose your heart. So I have a particular article for you to show about this particular thing. Okay. So this article was published recently only, friends. It was published in 2019. So they just mentioned in literatures and all, this mammalian zygote is described as totipotent cell. But when there is a molecular studies that they had done, totipotency cannot be a genetic totipotency. So here what they just mentioned means, so here they just mention zygote are retaining many key factors from the parental oocyte in order to reprogram an implanted differential genome, okay, or a zygote genome to a totipotency, like they just mentioned. And what it they say is that this particular totipotent, that is this particular zygote is not a true. So here you can able to see the mammalian zygote is not totipotent transcriptionally, epigenetically, and functionally also they are not totipotent cells, and also zygote is not a truly totipotent cell, okay, but it is totipotent genetically as any other diploid cell. But here they just ask zygote is a totipotent stem cells or not. Of course, only we know because zygote is giving rise to all the cells of embryo as well as it is also giving extra embryonic cells. Okay, they just mentioned, they didn't ask whether zygote is totipotent epigenetically, genetically and all. They just simply asked the option. Okay, so that's why many student has given this particular option. So if you check with the other option like CDX2 upregulate OCT4 none, this is absolutely a wrong option because the CDX2 downregulates these two transcription factor. Okay, whereas this option is also correct. 3 regulatory transcription factors. Of course, there are 4 regulatory transcription factors are there. Here they just mixed on one particular thing. Of course, this particular option is also wrong. How can they should tell this particular option? There are 4 regulatory transcription factors that are playing an important role in maintaining a pluripotency of the inner cell mass. Okay, so this particular option, that is zygote is the totipotent stem cells, is absolutely correct, and the cells in the ICM are pluripotent, that is absolutely correct. Okay, so option number A and option number B are correct. So those students who have selected option number A and option B will be getting an answer, getting a correct mark, and as well as those students who have selected B and C will also getting a correct option. Okay, so 2 questions for 2. If you attempted these 2 questions, then definitely you will be getting a mark. Okay, so if it's a 4 mark means they will be giving you 4 mark. If it is a 2 mark means they will be giving you 2 mark. Next question to challenge is question number 79. So question number 79 is with regarding to planaria regeneration. Of course, they just given these 2 options are correct. Of course, it is the correct option, friends. Here they just mention high level of ERK inhibit the head specification. Of course, high level of ERK, they don't have any particular role in inhibiting the head formation, friends. So that's the thing you should know. So if you check on this particular article where they just mention clearly about the role of this particular ERK. Okay. So here you can be able to see this ERK activity was required for differentiation of blastema cells in order to exit from the proliferative cells and to undergo differentiation. But what did they say is that ERK, they just inhibiting the cell differentiation like they just mentioned. So high level of ERK inhibit head space through high level of ERK, they generally what they will be doing means. So if you check on the thing I just explaining you, so when I was teaching you the unofficial answer key, I just refer you about planaria regeneration where I had given you a list of articles that you can refer, friends. So in this I just mentioned you clearly about the planaria regeneration. Okay, so you can refer this particular article, they had given you clearly about this thing. So when the Wnt signaling has been inhibited, head formation it will be promoting. So in planaria regeneration, the Wnt signaling will be inhibiting the head formation, they will be promoting a tail gene regeneration. Okay, the same time when you block or when you inactivate the SNOTM gene, it will be resulting the regeneration of tail in the place of head. Okay, so next thing is with regarding to this particular article. In this article also I just mentioned you to refer. In this particular article they just mentioned when you inhibit few component of Wnt signaling pathway, it will be causing a regeneration of head in the place of tail. That's why if you inhibit Wnt signaling pathway, there will be an animal will be having 2 head. Okay, so this particular thing you can quote. Okay, so this particular option is absolutely correct. So when the NOTAM is expressed in a posterior end, the planaria with 2 head will be formed, and when Wnt pathway is blocked or it is activated, the resulting planaria will be having a head at both ends. Okay, so these things you can't able to challenge. Okay, so the next question is regarding to 80. Okay, so the 80 questions also many students demanded, but it's absolutely correct. This friends, FLC is a strong repressor of flowering. At the same time, flowering locus D, they are involving in regulating the target gene for mediating a reprogramming of meristem to produce flower. Next, FT, it's a mobile signal, friends, that will be inducing flowering. At the same time, this particular FRA gene, they are activator of FLC. Okay, so if you want in-depth analysis with regarding to this particular thing, you can refer this particular pathway. Here they had given a complete thing about flowering locus 3, specifically about this particular thing. So here you can able to see there are 2 locus of FC, 2 loci regulating this particular flowering locus C. One is this FRAGIDA gene and another one is this Wnt3 gene. Okay, so the next question is regarding to question number 88. So this is, here I just mentioned which of the following option is incorrect. Okay, and the correct one is with regarding to 88 question is so option number A, that is ACD. Oxytocin and vasopressin are synthesized in posterior pituitary. Of course, it is a wrong option. Okay, they are synthesized from hypothalamus and they will be secreted in the pituitary gland. Okay, so here you can able to find out in this particular article they have given clearly about this. The oxytocin and vasopressin or neuropeptide that are synthesized in hypothalamus and they are secreted from posterior pituitary gland. Okay, so please don't challenge this question and all, friends, they are given correct only. Next is with regarding to C, that is alpha and beta melanocyte stimulating hormone are secreted from intermediate lobes of pituitary in adult human. No, of course, they are secreted from intermediate lobes only, but they are not being present in adult human. They are present in some other animals like rat and mouse. Okay, so here you can able to see this particular hormone is being synthesized from intermediate lobe of pituitary gland, which is a melanocorticone receptor antagonist, and this particular because human pituitary they lack this particular lobe. Okay, so that's why they had marked this particular option is also incorrect. Next thing is regarding to D. Growth hormone secreted from anterior pituitary is stimulated by hyperglycemia, like that they just mentioned. But you should know under the condition of this hyperglycemia, that is the hyperglycemia will be stimulating this particular somatostatin hormone release by hypothalamus, which in turn will be suppressing the growth hormone. But what did they just mention? The growth hormone secretion of anterior pituitary will be suppressing the hyperglycemia, but here they just mentioned they are stimulating the hyperglycemia. So that's why they just mentioned. So don't challenge this question and all. So next question is question number 1 or 2. So which of the following statement in relation to insect meat is not true? And I think for you this particular question also you will be getting mark, friends, because they just mentioned option number D is wrong. That is flies having a structure called frenulum which joins a forewing to hindwing. It is a correct option, but they just mentioned this particular option is wrong. If you refer this particular paper, so here you can able to see this particular paper was published, I think it was published in 2020 only. So here they just mentioned this particular structure frenulum that will be interlocking the retinaculum near the hind margin of forewing. The function of this particular frenulum is to couple the forewing as well as hindwing together during a flight. Okay, so for this question also you will be getting a mark. So for 3 questions I had suggested you will be getting a mark. Next is regarding to question number 1 or 3. Okay, so question number 1 or 3. So this is for this question, definitely I think you will be getting a mark, okay, because the India has estimated to harbor around 40% of global tiger population, like that research is saying. I don't know this particular option is correct or not. Whereas this particular option is wrong only, friends, because in India there are only 45,000 species of plants are there, and animal population is 91,000. But here they had given a ulta option, so that's why these 2 options are wrong. Okay, so for this particular option you can challenge, but I don't find any particular evidences to challenge this particular question. In ICUN database there was no particular data to challenge. Okay, so that's the thing. If you find any particular thing, then please do challenge, then please do tell me. And this particular questions are correct only, friends. Please don't challenge this particular question. It's absolutely correct. Regarding to interrogation, if you study this marker assisted selection in plant biotechnology or in applied biology, they will be having a detailed process of this integration where they will be crossbreeding between a species is mediated by repeated backcrossing. They will be repeatedly they will be doing backcrossing. So that process is called as integration. Okay. So next question 2 is just confusing for students. Is question number 117. So again, to find the answer for this question, they just mentioned both IgG fragment of antigen binding fragment and J chain are giving, but in nowhere I just find out this J chain are used as a secondary antibody in Western blotting. So in this particular Thermo Fisher website you can able to, so here they had given a list of antibody that has been selected for secondary antibody. So here they had given IgG whole molecule, gamma chain of IgG, FC fragment of IgG and fragment of antigen binding fragment of IgG. Regarding to IgM, they just mentioned IgM whole molecule and FC fragment of IgM and mu chain of AGM as well as light chain of immunoglobulin they just mentioned. But here they didn't mention anything with regarding to J chain. Okay. So please don't, you should answer this, you should challenge this particular question, because in the question from ENT they just mentioned these 2 options, that is both the IgG as well as this J chain will be used as a second chain, but nowhere they just mentioned this J chain is correct. So this particular option, that is option number 465 is a correct option. If you want, you can challenge by paying amount of 200. So next thing is regarding to question number 110, friends. So 110 question is regarding to this particular thing. So it is an absolutely correct option. From ENT itself they have given correct option. So don't challenge this particular question. The next is question number 155. So again, many student has demanded, when we had discussed the answer key, many student has says this is a wrong option, but this is a correct option. So coral snake they just exhibit mimicry, and crystal jelly they express bioluminescence, and African lungfish they express a process called estivation, and monarch butterfly they are expressing a aposematism. So don't challenge this question. Anyway, if you challenge, you are not going to get climbing for this particular thing. Next is question number 99. This is also many student demanded. This option is also correct. Previously we were discussing the origin of top, but here they are given region definition of domestication, absolutely it is a correct option, so don't challenge these questions at all. Next thing is regarding to 36. So when we were selecting this particular, so which of the following is typically a true of invasive species? They are selected only, friends. Just previously we were discussing it is a case selected, but invasive species which is an exotic species. Okay, so this invasive species will be brought to a new environment and they'll be competing with the native species for food. Okay, so this invasive species definitely they are selected species. So if you study the life history strategies, selected species, what they will be do, they will be producing a chief offsprings, they will be producing many offsprings, because that's why they can able to maximize the growth and they can establish themselves in a new area. So invasive species are selected species. So these are the little bit confusing questions and few questions that are being open for challenging. So if you want, you can challenge, and few questions listed on that you can't able to challenge. So please don't challenge those questions and please don't waste your money. So thank you, friends. If you have some other question that you want to challenge, please do tell me. For section 2, that is the 2 question paper I didn't analyze. Once I analyze, I'll definitely upload you what are the questions that you had challenged in shift 2. Thank you, friends. Thanks for watching this video.